Second week of development. Second week of embryo development. Second week of embryo development starts from 8th day to 14th day after fertilization. Major changes happening during second week of fertilization are changes happening in the trophoblast and changes happening in the embryoblast. Trophoblast is the outer layer of cells which covers the embryo. Trophoblast mainly differentiates into two layers. Inner layer called as cytotrophoblast and the outer layer called as syncytiotrophoblast. Cytotrophoblast is made up of single layer of cells. The outer syncytiotrophoblast is actually derived from the cytotrophoblast and is made up of multinucleated cells. In syncytiotrophoblast, many lacunar spaces start appearing. Spiral arteries directly open into the lacunar spaces and are filled by maternal blood, which is necessary for nourishment of the developing embryo. At about this time, the embryo has completely invaded the maternal endometrium and is closed initially by a fibrin plug, which is further replaced by endometrial epithelium. The syncytiotrophoblast produces a glycoprotein hormone called as HCG or human chorionic gonadotropin, which enters the maternal blood. HCG maintains the hormonal regulation of progesterone by corpus luteum and maintains the pregnancy. Till now we have seen some of the changes happening in the trophoblast. Now let us have a look at simultaneous changes that happen in the embryoblast in the second week of embryo development. At this time, the basal blastomeres of the embryoblast facing the blastocystic cavity delaminates to form a single layer of cells called the primary endoderm or the hypoblast. The hypoblast now migrates by proliferation along the cytotrophoblast to form the yolk sac. This layer of cells is also called as the Husser's membrane. The layers of cells of the embryoblast above the hypoblast are called the epiblast. Now, the embryoblast is bilaminar, consisting of columnar upper epiblast cells and a lower layer of cuboidal cells called hypoblast or the primary endoderm. The epiblast layer will form the entire future embryo. Soon, amnioblasts of the epiblast separate from the epiblast and form an upper single layer of cells continuous with the cytotrophoblast to form the amnion. The amnion separates from the lower cells of the pluripotent epiblast. Because of this separation, an amniotic cavity is formed. Summary. Trophoblast differentiates into cytotrophoblast and syncytiotrophoblast. Formation of primary villi. Changes in embryoblast. Formation of bilaminar disc with epiblast and hypoblast. Formation of two cavities, the amniotic cavity and yolk sac. Formation of extraembryonic mesoderm, splanchnopleuric and somatopleuric mesoderm. Formation of extraembryonic coelom. Formation of connecting stalk and precaudal plate.